What is this session, Emerging Technologies? Really, it doesn't mean anything. If I had my way, right, we would have called this functional genomics, but apparently that offended some of the population genetics people. Don't tell anyone I said that. But basically, I think this is, generally speaking, a lot of the talks in this session are going to be around sort of trying to infer function to some degree from sequences, right? They could be DNA sequences, could be protein sequences, lots of different variants in the types of computational approaches, but generally speaking, the general topics are in this space. Who are the organizers? Me, Harold, Ritambra Singh, who's sitting up in the front here, and Elior, who is also sitting right next to her. And so why is this important? If you have any ideas on what we should do in all the interactive sessions, you should talk to one of us. We're happy to take suggestions at any point in time. We have some ideas on what to do, so if none of you guys have any ideas, which is totally expected, no offense, we'll think of something. But anyway, come find us at one of the coffee breaks. We'll talk. Okay, so this is just a really quick overview. We don't have to go over it, but just to give you an idea of the types of things that we need you to fill time with, right? So there's a ton of talks scheduled, right? We have some sort of unscheduled time. In the schedule, it says something, right? Which I think no one really knows what they're going to do yet, for what it's worth. It's slightly a function of how many participants we have regularly. So today, I think because of the timing and stuff, we will likely do some sort of intro. We also want to get a feel for how many trainees want to give talks. So again, as you hopefully have noticed today, if this is your first CGSI is sort of geared towards trainees and trying to get them really involved. So we'd like to have sort of trainees be first and foremost in many of the sort of discussions and involved in many of the things that we do. And so one thing that I will do today, and my job as an organizer and other organizers as well, by the way, one of us will be here at any given moment in this session. So you can bank on that. But is really to sort of, one, make sure the trainees get first priority when you raise your hand to ask questions. So if you're a trainee, we'll try to do the trainees first thing. Obviously, you guys don't have to have questions. The other thing is that it's our job to slightly force you to talk, even if you don't want to. So I'm really hoping that everyone who wants to talk, and even those of you who don't really want to talk but probably should, give at least a 5-minute talk on something that you're interested in. So we'll talk about what that is in a moment. But just keep in mind, all these colored times, these are times that we have to fill. Last year in the interactive time, we ended up doing a sort of career panel type thing, which we can totally do again this year. We have time to do 2 panels, right? One that could be I don't know, I'm just making something up, work-life balance or whatever. But the other could be on different types of trajectories. One thing that I didn't really understand, at least in America, right? It's really common for folks like us to be either hired in an engineering school or in a medical school. And those mean 2 very different things. So if you're a trainee and you're kind of thinking about what life might be in I don't know, 5-6 years when you're on the market, happy to talk about things like that. Okay. And so the participant talks, again, priority will be given to grad students and postdocs, but literally anyone can talk. If you're a faculty and really want to talk, you should definitely email me and say that. We'll try to fit you in. I think we have time for everyone who wants to give a talk. But if you're a trainee and you're later stage, I don't know, think about maybe a 10-minute talk or something like that, 10, 12-minute talk. If you were very early stage, I strongly suggest you talk anyway, and we can do a 5-minute talk. And even if you don't have results, you can say, I'm thinking about diffusion models and thing X and applying them to whatever. Yeah, you should talk. Okay. And so it doesn't, again, need to be specifically on your own research, but the whole goal is to get to know each other, right? And hopefully, you see someone talk on something that you're interested in, maybe they're more advanced than you are, or maybe they're more junior than you are, and you go meet them at the coffee session. Yeah? Okay. Interactive time, we did a faculty panel geared towards trainees. We can do this again, or we can do something else. you tell us. Yeah? Cool. Any questions on overall logistics or anything like that? By the way, you can do whatever the hell you want. Like, you don't have to come here every day. Well, you're supposed to, okay, I'm not supposed to say that. You're supposed to come to CGSI every day. You don't have to come to this session every day, yeah? However, like, you know, there is some sort of like predictive modeling. If you kind of like this sort of thing, you know, you should sort of hang out. And there is some benefit to staying in the same sort of group, but you can go whatever you want. Yeah. So you don't have, you're not like married to this course. Okay. Great. All right. So I'm going to talk a little bit about some work that my group has been doing on hierarchical modeling. This is sort of the thing that we do in my group quite a bit. A lot of sort of small sample size with many, many parameters type things, right? And so if you saw me talk last year, this is sort of an extension of that talk, but with some sort of new results and maybe a slightly different twist on it. So last year, we were very early stage. This year, we have a paper out in the space and many, many more papers to come, and happy to talk on sort of extensions in that space. Okay, this picture is supposed to be funny, so you can laugh, but also you don't have to. But really, my group, like many of you guys, is really interested in like what is a function of some DNA. So you hand me some DNA, and we try to figure out that function. The focus of my group is maybe slightly different than what a lot of people in the predictive modeling space is doing. So we think a lot about perturbation style experiments. That is, you work with an experimentalist and they do something, right? They might give a drug, a toxin, something like that. They might mutate the DNA. They might actually insert some new gene, something like this, and then we see what happens, right? And these days, sort of the flavor in genomics is you do this in a very, very parallel fashion. And we're going to talk about that and some of these things in more general, right? And so why am I interested in this? What sort of things do we think about, right? We think a lot about all the different machinery involved, right? So there's lots of different steps involved in gene regulation, and one specific DNA sequence might have many, many different functions, right? And so in order to get at this, at least in some sort of very realistic way, right? Realistic there in scare quotes, you have to do experiments, right? At the end of the day, like, you have to do experiments. In fact, the weird crap that comes up in experiments is usually what you actually find interesting, right? Not necessarily what you might be able to predict like every single time in every single tissue, right? It's the weird stuff that is sort of interesting, at least to me, right? Okay, and so experimentally these days, there's lots of different approaches. The types of things that we think about in my group are usually of the following flavors. So we've been thinking quite a bit about massively parallel reporter assay. That is somewhere where you introduce a sort of putative enhancer, and then you have some sort of downstream assay where you're looking at what it could have regulated, right? We also look at gene-gene interactions through CRISPR screens. So we think quite a bit about what might happen when you knock out one gene and then measure some related phenotype, right? So this could be by flow cytometry. It could be simple cytotoxicity screen, which basically is a fancy word for like, did cell die? Yeah. And the thing that we're going to talk about today is what happens when you have a specific variant to a protein, right? So one specific point mutation, how does that actually affect the protein function? And function here is very fuzzy. What do I mean by that? It's basically anything you can measure, right? Today we're going to talk specifically about cytotoxicity. Again, this sort of dumb phenotype, which is like, did cell die? But a lot of the things that we're going to talk about actually relate to a lot of the related work that we're doing with flow cytometry and sort of other phenotypes that you might be able to measure. These days, really kind of interesting things are happening, and I think it's a really fun time to be in this space. This is kind of geared towards the trainees. Sorry. Why? Because like people are starting to mix like imaging and all sorts of really cool things, so you can like actually see like, oh, this mutation did this, right? And maybe I can get a transcriptional readout, but you can actually start looking at the morphology of the cell. This is sort of sci-fi-ish in the sense of like not totally there yet, but like there's been a few papers that are sort of getting on this. And I suspect that in the next like 5-10 years, someone's going to figure out how to do this really, really well. Okay. So generally speaking, we do computational methods for inferring function from cutting-edge assays, and we're going to talk about deep mutational scans. Okay, so biologically, why do you care about this sort of stuff? A lot of the collaborators that we work with care a lot about transporter proteins. So what are transporter proteins? They're basically proteins that move things, yeah? It's in the name, right? One such transporter is OCT1, and OCT1 is very, very important. Kathy Giacometti, who's one of our collaborators, has actually done a lot of really cool work on this protein, which is moves a lot of drugs among the cell membrane, right? So drugs that you might be familiar with, like metformin, right? Diabetes drug, and morphine, a pain medication, right? And with the wild type, like everything is fine, right? So you're moving across the drug along normally, everything is fine, we're happy, drug working, sort of as expected. But it's been observed that there's a fair amount of genetic variation in the population that unfortunately has a few sort of these loss of function or gain of function mutations where you don't actually transport the according medication appropriately, right? And so Kathy's done a lot of really great work on OCT1 along with a number of her collaborators, sort of showing this both pharmacologically as well as like in a large population scale wise, right? And so if you're interested in this sort of thing, this paper is now published from Kathy and Willow and all these other folks. You should definitely check this paper out. They did a really, really nice characterization with a lot of follow-ups and a ton of different assays. But a lot of this sort of stuff, the foundation was done using deep mutational scans, right? So they did a ton of different genetic variants with basic cytotoxicity screens, and then used this to sort of nominate a bunch of other assays that they were doing downstream. So that's kind of how the technology is used, generally speaking. There is a fair amount of interest in deep mutational scans in the sort of rare variant space, as you might imagine, because you can actually make every single possible mutation, and you can actually take some sort of measurement, right? And so how do you do this? I'm not going to talk too much about it this year, but there's a lot of different ways to do deep mutational scans, at least generating the libraries. And the way that Willow's group does it, Willow's one of our main collaborators, they developed this technology called DIMPLE. But basically, it allows you to order a bunch of specific small oligos and then do some Golden Gate cloning or Golden Gate assembly to basically create a library where every single variant, right, including insertions and deletions, are sort of represented in that library. And now you transfect this into a bunch of cells. In fact, this technology, it has a specific cell line that has what's called a landing pad, and you insert the gene directly into the DNA, right? And so it's sort of expressed in sort of a natural environment. It's obviously like not natural, but like in sort of about as close as you can get in one of these high throughput experiments, right? Okay, so if you're interested in the technology on how to actually do this, you should totally read this paper. It's in Genome Biology 2020 by Christian McDonald. Okay, but the bottom line is you end up with this library where you've created every single genetic variant to amino acid, right, of a specific protein, including every single synonymous variant, every single missense variant, including insertions and deletions. Now, technically speaking, you can do pairwise mutations and all sorts of stuff like this. We're not going to talk about this today. It's not sort of the regime that we're working in. There is some preliminary work on doing this at a much larger scale. Happy to talk about that at coffee breaks if you're interested in. But for today, let's talk about point-wise mutation to amino acid, right? Okay, and you can think of the sampling being done as effectively, you integrate your library at one point, and then at some specific time, hopefully an early time point, you sample, and then you sample again at a later time point, right? And what does sampling mean? You sequence the entire library, right? So what does the sequencing get you? It gets you the relative representation of every single variant and how well it's sort of represented in that population. And you sequence again over and over and again, right? And so what you end up getting, right, is effectively for every single one of these possible mutations, so in this case it's like position 2, a G went to a P, we got this many counts, and it's a big matrix, yeah? And you'll end up with effectively what some of these folks call as fitness curves, right? And you want to infer this fitness curve, right? So if we're talking from a linear regression space, the slope and maybe the intercept, right? And some uncertainty associated with it, right? But the key point here is actually that you don't have a lot of data. What do I mean by that? While you do have a lot of different mutations, you have like 2 to 3 replicates per time point. This is bad, yeah? Any of you who have done statistics, know this is a bad place to be in. Yeah, this is inherently a futile exercise, right? So how do you get around this? So this is sort of the trick and sort of the hat trick that we'll do today. Okay, and so just to sort of set this up, naively, what happens? Well, what we want to just sort of understand is a slope different than what I expect under a control. You can think of synonymous variance for today as sort of a control, a negative control. It actually isn't totally true. There's some interesting work showing that synonymous variance actually might behave differently in different scenarios. But generally speaking, they should have a significantly smaller effect than a missense mutation, right? And you need to infer, let's say your protein is about 200 bases long. Let's say 4,000 slope parameters from 3 replicates. Kind of a bad situation to be in, right? And all the uncertainty parameters, right, which on the order of 8,000. Bad place, right? And so unsurprisingly, as you might expect, you can end up with a lot of false positives and be very underpowered if you're doing sort of the naive t-test, right, or just fitting a standard linear regression in this space. And so how do we get around this? Well, there's a lot of different ways to do this, right? If you're familiar with DE-seq, for example, DE-seq is a very general term, a general method. What do? They regularize the variance as a function of the mean, right? We do this, right? In fact, it's under the hood here. I'm not going to talk about that today, but in addition to that, you can inject some sort of domain knowledge, and this is basically the hat trick that we use all day long in my group, right? So we think really, really carefully about all the parameters that you're measuring and all the things that you actually care about and what the experiment is supposed to be doing, right? How are these parameters related? And then we put some informative priors on those, right? And so in my group, an informative prior is actually not a bad word, but it might be to some of you, and that's okay. I'm happy to fight about it later. So how do you sort of think about this, and how do you motivate this? So we get this from actually having a number of different observations, right? And these are very biologically motivated, because turns out, I know we're all computational people here, but turns out that proteins are 3-dimensional structures, yeah? They're not just a line, yeah? They're not just a single sort of sequence. And because of that, when you mutate one position, it actually affects the whole thing. Weird, right? And so, as you might expect, certain amino acid substitutions actually behave somewhat similarly, right? And so we see this, right? And so one observation you might make is that actually mutations at a specific position are correlated, right? If you can knock out a specific position or change the variant at a specific position, you might not observe anything, right? And it doesn't matter what you change it to. But other positions, you might change it to something, and no matter what you change it to, you're screwed. It totally breaks the protein, right? So that's the first observation. This is actually a pretty sound, biologically motivated observation, right? The other thing is that you might observe that at some positions, they might be correlated, right? The amino acids might be correlated in their behavior, but some of them actually behave fundamentally different, right? So maybe sometimes I substitute an L, and the L and the V, as you can see here, the L and the V and the I sort of behave somewhat similarly, but then they behave very dissimilar to the A, the T, the S, and the Q, yeah? So this is sort of kind of expected, and we'll talk about that in a moment. The other thing is that different positions have sort of different scale of effects, right? That is, you might expect a much stronger sort of effect at some specific position, or you might expect a weaker one, right? And on average, the sort of observation turns out to be somewhat true. Yeah? So hopefully these are sort of clear from this picture. Okay, so the first observation actually, if you sort of just take the mean, this is posterior mean, but it kind of looks the same, at every single position, variant effects, right? Sometimes I'll say variant when I mean variance, and sometimes I'll say variance when I mean variant. I'm sorry. Just impute the right one. Okay. So you do see that actually, and again, this sort of makes sense, right? Like in some positions, like there's very strong domain-specific effects. That is, like this whole chunk of the protein, when I mutate something, it's gonna, like, it doesn't matter, right? Like here, in some, if we're using statistics terms, they're somewhat exchangeable, yeah? Okay. And so we wrote a paper on this. This is part of the model. This isn't the whole model, but this is part of the hierarchical model. And there really isn't, there's tons of other parts of the model, but this is really the sort of core idea here, right? We have some specific variant that I care about, right? The variance score, right? This is the slope, that's one specific slope. And what I do is I put a position-specific prior with a position-specific variance. These are sort of, there's some additional hyperparameters that are shared across the entire experiment, but this allows us to basically shrink things relative to the, shrink the betas relative to the specific position, right? Okay, this could be very dangerous. In the paper, we argue that it's not, under a number of different regimes. I think we do a decent job about sort of justifying that, but you know, we can argue about it later. But for those of you, this is particularly geared towards trainees who haven't really thought too much about Bayesian estimation. You could kind of think about Bayes as being sort of this tug of war between the prior and the observations, right? And so why do you care about doing this, right? Well, I guess let's talk about this picture on the left first, right? So if you don't have a lot of data, you can sort of think about weighing the prior and the observations to some degree, right? But as you get more and more data, the goal is to sort of get closer and closer to weighing the data more than the prior, right? And if you've sort of taken classical statistics that has looked at some of these things, some of these classical Bayesian estimators, you'll see that like with infinite data, right, the MLE and the Bayes estimator sort of coincide, right? Okay, so fine. But why would you do this, right? Well, if you are working in this world where there's not a lot of data, there are some properties that one might want, right? In this case, what we want is actually lower variance over all of the experiments on average for a little bit of bias, right? And the art of this is really trying to inject the least amount of bias while still reducing the variance greatly, right? This is kind of hard to do, yeah? It takes a lot of practice and a lot of sort of model checking and all this sort of stuff. We're not going to do any of that today, unfortunately, but we've done this, yeah? Okay, so generally speaking, conceptually at least, Rosas, this method that we have this paper on, is sort of like this, right? There's some position mean that is sort of weighing you, and as you get more and more data, you sort of move towards that more and more. Okay, fine. We end up with lower false discovery rate while maintaining power. So for those of you who kind of like the math, we don't do Gibbs sampling, but you can take a Gibbs sort of view of the posterior, right? M is the data here, and you know the rest of it is hyperparameters. And so the thing that we care about here is this beta, and the posterior under a Gibbs sort of view of the model looks something like this, right? And so this is the position-specific variance that you're learning actually through some degree of sharing. This is the sort of mean variance relationship that you might see in something like DE-seq. And this is the positional mean, right? And this is the data, right? And you see that there's some sort of reweighing as a function of these variances, right? So if at a specific position you have very little variance, that is, if no matter what you knock out with, it actually like will always screw up the protein, then you're going to weigh the position more than you're going to weigh the data, right? It's just sort of how it works, right? Okay, I'm not going to spend too much time talking about this, but because we're running later than I expected, actually. But okay, all right. So what's this sort of new thing? What is the new thing that I want to talk about today? So the paper itself only focuses on the first observation. And some of the things that my student Jingyou Rao has been working on is really how to deal with observation 2 and 3, which turns out like dealing with this a little bit in a more general sense is quite complicated. And we can talk about that later if you're interested. I don't unfortunately have time to talk about all the sort of specifics. But how do you sort of deal with different sort of variants that sort of behave similarly, right? So we can turn to like evolution and sequence alignment for some inspiration, and in fact, that's what we do, right? So the blossom matrices, which many of you folks are familiar with, effectively provide some sort of weighing of based off of evolution and a bunch of previous fit models, right? On how likely a substitution is. That is, if you move from a C to an E, how likely is that relative to something else, right? And more positive means it's more likely. More negative means it's less likely, right? And so what we do is we add another parameter into the model, right? This new parameter, NU, right, as a function of the specific variant. And what we do is we have now a bunch of variance parameters that are parameterized by this blossom matrix, right? That's the intuition, right? And the way, conceptually at least, how you might think about this, right? So this gray bar here might be the observed sort of variant score, right? Something that you might learn directly from the data. And this is maybe the positional mean, right? So the positional mean is sort of the average of everything at the specific position. And you can think of this new parameter as effectively scaling based off of different groups, different types of amino acid substitutions, right? And so it'll get you a bit closer, and again, injecting a little bit of variance, a little bit of bias for this trade-off in variance, right? And so this sort of gets you partway there, right? And this is learned on average throughout the entire experiment, right? So even though we're adding a ton of parameters, seemingly so, we're shrinking across the entire experiment also. So it's not effectively as many parameters as it looks like, right? However, we talked a little bit about how every position might have a relative sort of scaling, right? On average, the position mean isn't going to look the same, and the data sort of shows that, right? And so the way that you get around this is actually adding another position level scaling that enables you to sort of linearly scale this new parameter, which we call rho. And this is now saying that we're going to scale the amino acid substitution matrix some amount more or less than other positions, right? Now, we have a bunch of extra stuff, right? Right. So yeah, that's what it would look like scaled. We have a bunch of extra stuff. If you look sort of carefully, right? So previously, beta depended only on this parameter phi, right? And now we're adding this other thing, nu and rho. From the learning side, this turns out to be a really, really annoying problem. It's actually, if you look carefully, it's unidentifiable. So you have to make some constraints on it. We could talk about that later if you're interested. But the more interesting thing here, I think we figured it out, yeah? The more interesting thing here, though, at least to me, is that now you have two different new parameters, NEW, that are telling you something about the position, right? That are effectively reweighing themselves, yeah? So we have something that's my position score, and also how much sensitivity to the amino acid substitutions I have, right? And so this is kind of interesting because I can create summary statistics, or effectively summaries of the posterior, that will tell me what this looks like, the position score versus the positional mean score versus the amino acid sort of contribution, right? And so a priori, I might expect a bunch of different sort of combinations of these two things, right? This would be, it might look something like this, right? If I were to plot across the entire protein the position score versus the amino acid sensitivity score, it could look something like this, which means that there's effectively no amino acid sensitivity, right? Could be a thing, right? It basically, anytime I break some specific position, it breaks it. It doesn't matter what the amino acid is, right? The other thing I could expect is something like this, that at sort of really strong gain or loss of function, I end up with some with very little amino acid sensitivity. But when there's sort of more neutral effects on average at some specific position, I might be more sensitive to some specific amino acid position. Substitutions. The other way to think about this, there's obviously many different ways, right? But another sort of hypothesis that you might have is that for some specific, for any sort of specific score, we have this really wide range, right, of amino acid substitutions, and it really depends mostly on that. Okay, so what does it look like when we get real data? It turns out that at least all the data sets that we've looked at, which is on average, or not on, I think we've looked at about 10 so far. This is still like very, very preliminary. They have this sort of U shape, right, which is this sort of regime, right, where the position, at a really strong gain or loss of function, position dominates, but when you're closer to neutral, the amino acid substitution matters more, right? Okay, so why is this interesting? Well, it turns out that you can actually look to see, effectively, this is doing some sort of like latent clustering, if you will, on how the different positions might behave, right? So there are some positions, and we can identify these now from looking at some of these plots, that you see this sort of mixture, right, of amino acid substitution effects, and in fact, some of them look very similar to each other, and that's sort of expected because different amino acid substitutions have different charges, and so on and so forth, right? Here, at really strong gain of function, right, doesn't really matter. Really strong loss of function, again, really doesn't matter, yeah? Actually, this is a weird protein where it's sort of flipped in the score. But yeah, so you can look at a lot of these different sort of plots and start getting this. Now, we're still in the early stages of figuring out how useful this is going to be, right? But it's sort of now providing the experimentalists another way to sort of summarize this data, right? Because what would you do before this? You might do some k-means clustering where you end up getting like a ton of different a ton of different clusterings. You might try to do some more complicated sort of latent model approach from like take your flavor of ML and sort of do this. But this is sort of using, at least to like a first order, some experimental driven knowledge, right, of how all these parameters are related. Anyway, so bottom line is that the way that this sort of, what we've done so far, today at least, is sort of implement this new model, which extends this previous framework. We're actually trying to see what we can learn from these amino acid substitution estimates. We're not done yet, but sort of to whet your appetite a little bit. We've been trying to, one thing that we've noticed is that these sort of the variance in these scores actually seems to be fairly well correlated with alpha helices. And what we're trying to do right now is see if we can actually predict the secondary structure directly from these scores. And it sort of looks like we probably can do just as well as you can with all the sequence information, which is kind of nuts. So I don't know how well this is going to work. This is still like early stages, like as of last week, which is why I don't have slides on it. But we are thinking quite a bit on extending these models. We have, actually related to Joe's sort of talk earlier, we have quite a bit of work in the pharmacogenomics sort of space where you titrate drugs and you see, or other sort of things that you might care about, and actually ask when you have like, in pharmacogenomics, it's called EC50. This is the like midpoint, like mid-saturation point. So you can actually do this now in a very systematic way using flow cytometry. We also have another student who might be here, Joanna Jiang, who's working on modeling the three-dimensional structure actually into these estimates using a Gaussian process. And so we have I have a lot of opinions about deep mutational scanning, a lot of inference problems, and so happy to chat further. If this sort of space interests you, this YouTube channel, CMAP segs, I don't know, whatever. They have like even, look at this, is such a sad automated 8443. Maybe that means someone to someone, but just look CMAP_CEGS, and there's a ton of, turns out there's a lot of people who care about deep mutational scanning, and there's not actually that many computational people working on it, relatively speaking, yeah? And so my student who actually primarily, this is most of the work here is hers, Jingyeo Rao gave a really great talk there a few months ago called What is an Invariant Score? And so it gives a high level overview of like the actual statistical problems there. Okay, so just acknowledgements. The folks in my group who are primarily working in this, Riley, who's actually going to Chicago. She was an undergrad in our space last year, but this work was primarily done by Jingyao Rao, and Jerome has been working on a lot of the facts-related stuff. Folks at UCSF and independent folks who've been working with us. So happy to take a question or two. I know we're way over. Sorry, I won't eat into your lunch anymore. Yeah, thanks.